So thanks for coming. Welcome to the second colloquium of fall 2023. Fall. I'm very excited to introduce Doug Shepard. He is an assistant professor in our Department of Physics at ISU. He moved his lab from CU, University of Colorado Anschutz in 2019. Doug did his training, undergraduate training in physics at UC Santa Barbara. And then he took a pause, he took a sabbatical, or he took a break from science. He was a professional climber. I'm really impressed by that. I'm not sure whether you still climb, but I know you hike. I saw you outside my house from time to time. I live near South Mountain. And then he continued maybe, seeing beautiful scenery when life is too boring for Doug because it was long before the pandemic. Van life was nothing at that time. He continued his training at CSU, Colorado State University in physics. And then he received a nice fellowship, prestigious fellowship from his postdoc at Los Alamos National Lab. One thing that I always admire and grateful is that Hao is very kind. He's a good mentor. He and I are co-mentoring a student. So the story was he was kind of frustrated by the approaches that are available to him in my lab. So he approached Doug, and Doug was not only say yes, but he was fully invested in the student success. I mean, I think you got what, 2 COVIDs from mentoring Frankie?
Oh, I don't know.
Probably just one. I know for sure he got COVID because of my student. And I'm grateful for that. With that, Doug, take it away.
Great. So thank you, everyone. Thank you, Rizal, very much. So can people hear me in the back, or do I need to turn it up? All right. So we're going to have a little bit of a whirlwind today. I'm going to try a demo. We will see how it goes. I'm worried I'm going to set my computer on fire because I'm going to try and run a camera of a robotic micro swimmer once we finish our introduction. So we'll see how it goes. But to start, I want to give a little bit of kind of theoretical footing on something that I didn't even realize I was doing as a graduate student, which was I was using fluctuations to study sort of the natural world. And in fact, it's really only been through here and new projects we've had with Rizal and actually with Peter Brown, who's a postdoctoral scholar in my laboratory, that I've really come to more appreciate how all these things tie together. So I'm excited to tell you about some of that work today. So we build microscopes as part of what my group does. And the reason we do that, and the computer may have already died. There we go, is to study biological systems, both biophysics, sort of molecular biophysics, but also sort of things that are scaled up a little bit, more complex. So what you're seeing here is actually 3D cell culture imaged at sort of near video rates in 3D. This is actually not an easy feat to achieve, especially in multiple colors using a fluorescence microscope. In particular, the little dots inside of here are little 100 nanometer vesicles that are inside this that are part of the recycling system of the cell, which are called autophagosomes. It's unimportant for the rest of the talk. We're not going to come back to it. The point, though, is we have the ability to image sort of at the diffraction limit of light in 3D very fast due to some of the advancements that we've made in my lab and working with collaborators. And today I'm going to tell you how we use that to study sort of a more fundamental biophysics problem that has to do with fluctuation dissipation and Brownian motion. So to do that, I want to come back and start, with Robert Brown, who was actually a botanist. And so he was involved with maintaining the Royal Society's plant specimens. And as part of this, he would often use a microscope. And in the courses of doing that and actually just sort of working in all sorts of different areas, he noticed that, things under the microscope sort of, to put it the way Oliver put it, sort of jiggled and wiggled, right? Things moved around. And at the time, you have to put yourself back at the time they were doing this work. We didn't have the sort of concept of the atom yet, right? We still thought matter could be sort of continually broken down into smaller and smaller and smaller and smaller and smaller pieces. There was no discrete idea. And in fact, even kinetic theory and the second law were being hotly debated at the time. So we're really in sort of a cusp of a change in the way we thought about microscopic physics, particularly statistical physics. So it's not an academic paper, actually. It's a letter that then got published and passed around where he describes some of these original observations that he had. Then he went on and he did a huge amount more observations, which aren't really well understood. And actually, he was misunderstood in this original paper, sort of attributing these objects as being alive when what he was really trying to wonder was, could there be something like an atom or a molecule bouncing into them? So this result sat for a while and people debated it, but actually it wasn't that interesting until people started really getting more evidence that this is true for inanimate objects. And then that, it wasn't some actively driven process of life that this wiggling and jiggling was happening. So if we take a step back from the physics and the biology, if I watch these bounce around and I plot their displacement, what I'll eventually come up with is something that I think a lot of us in this room have thought about or maybe heard about, which is called a random walk, right? So this is described in a lot of different ways, but the idea here is if I have some process that's resulting in a chance and let's say moving left to right, or in this cartoon from Gemov, moving in these different sort of moves, I'm going to get a statistical distribution of where I end up from where I started. And that can be really well described and it's highly repeatable. So in fact, this is sort of a predictable randomness in the sense that the final outcome is drawn from a probability distribution that is always going to be the same. It's just any given random walk is a realization of that distribution. And so there also was work going on at the time about thinking about what does it really mean for these statistical quantities to play into how we thought about thermodynamics and sort of statistical physics. I really want to drive home that these statistical properties of the random walk are general, and in fact, they don't scale in the way one might think they will. So if I was to walk in a 1D line, I'm going to visit every site, with some probability. If I do it in a 2D, actually, I can still visit every site, but actually in 3D, if I was to bounce my way around this room like a gas molecule, there's a chance that we block this into XYZ coordinates, that I would never visit a site there, right? So this has pretty serious consequences for the way we think about life and sort of physics occurring if we're saying these microscopic ideas drive the way statistical physics happens, right? But we don't necessarily, visit all of these. And so I think this is a really important idea that we often trivialize this to this 1D case when we're teaching about it. But in fact, in 3D, this is a much more complicated sort of process. And this is a general outcome for any sort of statistical process that follows this. We haven't really described Brownian motion yet. So 3 sort of key physicists thought about this problem. And so, in fact, this wasn't done in a vacuum. I've just highlighted these 3, but in fact, there's quite a few other people who thought about this at the time. So we have Albert Einstein, Marian Smoluchowski, and Paul Langevin. And actually, Paul Langevin's paper came afterwards, but I'm including him here because I think the result is important. And so Einstein had this, miracle year, right, where he published all of these results in 1905, and Brownian motion was one of these, where he actually made a connection between the sort of Stokes drag of a particle, right, and this sort of observed random motion and put them together to give a prediction that could be testable. It turns out that independently, Smoluchowski had also derived something very similar, but more from first principles of particles bouncing into each other. And then Langevin came along later, and actually this is one of the first papers where this is sort of famous Langevin equation, and we have this solution for how the particles are moving around in the solution. And so the key point here is we're going from what is a random process, right? You don't need to understand anything about physics to watch these random processes occur and see that you get coin flips that average out to 50-50, right? You can do that without any knowledge of the underlying physics. But if you think about the problem carefully and think about why is this occurring and how do I infer something about the system, what you can actually do is start to explain some of the confusing properties that Brown had seen and other people had seen investigating it. So one, the properties of these solutions, which are again, particles inside solution, they change the particle type, they ground up animals to do this. One of the things was an Egyptian sphinx, pieces of that. They put them in solution and they said they don't change. So wait a year, I get the same statistical properties. You can change the viscosity of the solution. So I have a whole vat of corn syrup here, and anyone who's come by my office lately has actually heard me using a drill trying to mix this with a paint mixer. And the reason for that is to change the viscosity. And that says that you get motion changes with the viscosity of the solution. So there's something about the viscous drag that's changing the observed random motion. And then also it changes with applied heat. If they applied heat to it, they got sort of larger displacements. So what is the physics that was underlying this? And the real question came from Einstein's proposal is that, this is coming from this interaction with something much smaller that you can't see inside the solution, right? In this case, it would be water molecules, let's say. And there's many collisions, and those collisions are happening very fast. And in fact, the instantaneous velocity is sort of an ill-defined idea. And in fact, even today, And there's a lot of really nice review articles written about this, even up into sort of 2016, 2017. No one had actually watched the instantaneous change in velocity of these Brownian particles. It's taken quite advanced experiments running in the megahertz to see this. It's sort of a version of precision physics that's done on this Brownian motion. So the question is, if this is true, if we have lots of different, collisions, on average, they're sort of giving us some displacement in the motion. A, is that true? And so what you could do, you could try and make it quantitative, and you could ask, what are the observable consequences, right? So what are the observable consequences to this deep connection between the fluctuation that's occurring and whatever is happening inside the sample? So this equation is that result as presented in the original papers. So what this says is, this is really what Einstein was thinking about here, is this is a way to predict Avogadro's number and sort of get at that, but also test that atoms are real. And so what this is telling us is that the mean squared displacement of the particle is related to something about the sort of thermal energy in the system and the viscous drag of the particle. And so this is how it was originally presented. There actually was a difference of a prefactor in Smolakowski's original paper. Langevin came later and sort of put this all together. So to summarize where we're at, again, you have to we take these things for granted now, right? That we have this sort of statistical view of the second law of thermodynamics, or maybe you think about fluctuations or Brownian motion. But this was really challenging the world at the time, right? I mean, the sort of atom hypothesis had not been accepted, right? And so this was a way that this was tested. And this is why this is so important, because it gives this connection. So how do you test it? And now we're going to get into the sort of philosophy that my group is trying to extend with. So Jean Perrin published a paper in 1908 and then actually did some more work. This is a translation I could find, which came later, where he took a microscope. They didn't have cameras then, right? So what did he do? He had this sort of version of a viewing glass that he could both see what was going on in his microscopic experiment, and a piece of paper where he hand drew the trajectories, right? So keep that in mind, right? We're going to hand draw the particle trajectories. So this is an example of one of those that someone digitized. And then Langevin made the really intelligent, observation that what you should do is then retake these and plot how far away were they from where they started on some equidistant circle to ask what's the mean squared displacement. And once they went through this, they actually found that, sure enough, they got an incredibly accurate guess for Avogadro's number the first time. And actually, they were able to validate that this hypothesis was correct. And so this didn't solve the conflict, right? And so there's a lot more history here, but the point is that this experimental verification took this combination of the fluctuations in the system, some thought about the physical model, and then some advances in the way that the experiments were done to put that all together. So I find that really inspiring, because it took a really simple result and it learned something, quite complicated about it. And what it comes down to is, this is a form of the fluctuation dissipation theorem. So this says us that the fluctuation that we observe, right, is due to, in this case, the viscous drag, the dissipation in the fluid. And so we can think about this as a transport phenomena, right, where we have some force that's being applied to the sphere. That's going to have something to do with the velocity that the sphere is moving at and something about its drag coefficient. Or we can think about this in the dual view, in the view that Einstein told us, which said, well, look, that coefficient, right, should be proportional to something about the thermal energy divided by the diffusion constant, right? How likely is it, do we think that that thing's going to bounce around? And so this dual viewpoint is an incredibly powerful tool in biophysics, because everything we're interested in really is in this regime. And as Oliver described last week, this really drives the ability of cells and molecular machines to work, because that amount of KBT that's there is not that much. All right. So what do we do? So we're broadly interested in a few areas. The main project I'm going to tell you about today is this project, where we're thinking about what the propulsion efficiency of a molecular propeller is. And actually, this is the student that Rizal mentioned that we ended up co-mentoring. This is a project that he and Peter did together. But for a long time when I was at the medical school, I really worked on how do we take these ideas and apply them to complex, actively driven systems that seems like this shouldn't work, right? So we really thought about, how do you do this for gene expression is what it comes down to. And there's a lot of interesting observations in the literature about gene expression, that it follows these same predictable randomness. The underlying physical model is much more confusing, but it turns out that if you make observations over time of cells expressing genes, and you do these statistically, you actually get things that look like repeatable distributions. And I found that very surprising when I started working in this field. And then more recently, we've gotten interested in how do we extend this idea of the hydrodynamics we were doing into actually watching bacteria swim at sort of thousands of volumes per second with new microscopy. So we'll see how the demo goes, and if it goes well, we'll talk about the new microscopy. If not, I might set someone's motor on fire. All right, The point of all these fancy movies is they can be quantified. So that's the other thing my lab really prides itself in, is that we don't just make microscopes to take movies, we actually think deeply about what the underlying physical model is, right? So for this microscopic propeller, sort of paper I'm going to tell you about, we actually wanted to learn something about when this thing rotates, how much does it move along this long axis? That's going to tell us something about its propulsion efficiency. For the gene expression, we think about how do we build actually sort of physical models that are interpretable. They don't have every chemical reaction in them, but it turns out they're predictable. And then finally, we also think about how do we interpret those very fast hydrodynamics in the context of confined environments. And I'll come back to that at the very end. So I'm going to gloss over a huge amount of work, because as Rizal said, the lab got started a while ago. And so I just want to give you 2 slides that kind of summarizes a lot of effort on a lot of people's parts before I talk about this one specific project. So one thing we've done in my lab is we've really thought about how to engineer the physics of light as it interacts with matter, in this case, biophysics samples, to generate sort of on-the-fly fast 3D imaging by manipulating what are called light sheets. And then also we use what are called spatial light modulators to manipulate the light that goes into the sample and learn new information that's coming out of it. So I'm not going to run through the details of how these work. I will talk a little bit about this in the context of the Propeller project, but I do want to say we've been really successful at this. It's still going. One thing we really try and practice is open science. And so we tend to not patent these things, and we actually tend to try and help people build them, right? So we bring people to the lab, we show them how to build these scopes, we distribute our tools for free, and we're really on the bent of let's help other academics or even companies do these experiments better. This idea that we can take these fluctuations and apply it to complex, systems that aren't immediately clear fluctuation, dissipation works, that's also been successful beyond what I possibly really thought was going to be, go. And during COVID, we got involved in trying to understand the gene expression that occurred during the virus entered into particularly cells inside the lung. So we have this series of papers with collaborators. It's what's called the Human Lung Cell Atlas, and also part of what's called the NIH Brain Initiative, where our tools, our molecular and our imaging tools, have been used to actually probe the locations of RNA during infection, ask things about what's going on at these, both in healthy and diseased tissue. So we spent a lot of time thinking about how to get these tools sort of scaled up and image millions of cells with sort of single molecule resolution. So the tools I'm going to tell you about today can go to that scale, but we're going to focus in on like a much cleaner fluctuation dissipation story today. There's people's names who do all this work are at the end. I just want to say a lot of people put a lot of hard work in this, and they're not going to all be represented, through this one thing I'm going to talk about. So I'm very thankful to everyone that's put a lot of effort in. Okay, so bacteria swim. And they swim in what's called a low Reynolds number environment. And so what that means is it's incredibly viscous. There's a huge amount of drag for them. And I was talking with Navish. Actually, there's so many people here. I don't know if he's here or not. But they stop, like you said, in angstrom, right? So if the motor stops and they try and coast, they stop within an angstrom. That's crazy to my brain. There's no inertia, none. Right? And that's a really weird environment to think about how do you move. I mean, if you think about you get in a pool, you can even imagine getting in a pool of pretty viscous water and you could still try and figure out how to move, but you might not coast. So what I want to tell you about is, Navish, as we know, recently joined our department, he's thought a ton about how the motor works, and in fact came from the lab that has really pioneered a lot of these studies, so Howard Berg's lab. What I want to talk about is how do we think about what the efficiency of that propeller is. And I want to do it from this dual viewpoint. I want to do it from the fluctuation dissipation viewpoint instead of trying to actively drive this propeller. But one could think about trying to build a macroscopic model of how this works, right? And so what would you need to do? You need to sort of look at the sort of span of life, and you need to think about what this Reynolds number that I just told you they live in low is. So the Reynolds number tells us something about the density of the fluid we're in, the speed we're moving at, the size of our object, right? Sorry, the density of the object, and then the viscosity of the fluid. So all of these small swimmers live down here where these Reynolds numbers are significantly less than 1. And we and large fishes and other things actually live, sort of whales and also dolphins live in this regime where you can exploit other ideas like vorticity, right, and turbulence and things like this. So if we want to put ourselves in the mind of how do we scale up to think about this problem, we have to think about if we made the object bigger, what would we have to do to make our sort of test trial be the right Reynolds number, right? Because these things, if we're just saying we just need to scale with the size of it and density of it, then we should just be able to scale the viscosity. So Evan Ozkan spent part of the summer with us thinking about how to do this problem. And so it turns out that if you want to make a macroscopic robotic version of an E. coli, which I'm going to dunk in this vat of corn syrup in just a second, then you basically have to use like 95% corn syrup to get to the right viscosity. And even then, it's not quite right? You're in a Reynolds number of something closer to like 0.1, right? So actually, it's not going to quite perfectly behave. And part of the reason here is I don't have a big enough fish tank. So we'll go from there. So what we're hoping to see is something like this beautiful set of movies that were from Asher Shapiro in the 1970s from MIT, where if you took a swimmer and you took this strategy where we have a reciprocal swimming motion, we're not going to go anywhere because there's no ability, right, for that inertia to go. The other way to think about this is we're in a regime of the Navier-Stokes equations, where if we have this sort of thing that looks the same forwards and backwards in time, we're not going to generate any ability to have propulsion. But if we take something with a tail, then what we can do is try and put it, in this solution and get that to work. And it'll actually start to swim because that's not a reciprocal motion, right? As the helix turns, if I brought it backwards, it's not the same helix anymore as it was where it started. So we're going to try this with the macroscopic one. We're going to talk about what we'd have to do to scale that up to be a real experiment. And I'm going to give you my argument why I think it was a better idea to do it on the microscopic level. So while you can kind of get a sense of scale from here, and I'm going to try and dunk this thing in the corn syrup. What is true is that this is not a unique solution for this helical propeller. There are other solutions that nature has come up with, and those solutions have to do with, somehow still beating this reciprocal motion idea, but doing it in such a way that they're moving in slightly different way than this is. So let's find out. Is it going to stay? All right, it looks pretty good. So what I've got here is I have a vat of corn syrup. And what I put in it is I put in a mechanical little robot. And what I have looking at it is a camera. And then I've shaped a laser beam into a sheet of light. And so then I have a remote. And again, Evan built this. And so now what we're going to do is try, I shouldn't have taken the glove off. I have to get it in the field of view. Is we're going to try and start this thing and see if we can see something about what the fluid dynamics look like around it. And then we're going to use that to think about how can we understand what the propulsion looks like. So it's turning. All right. So let's see. There we go. So here is our propeller. And I hope everyone can appreciate it. Let me drag it over here. Can everyone see that the fluorescent beads that I have inside of there? And some of these are air bubbles. They're not all beads. I mixed this really quickly. And again, when you mix with a drill, it's not the best situation. They're not moving very much, right? We have that viscous damping, right? Remember I told you we're not quite at the perfect Reynolds number. So then if I come and I bring the swimmer, into where the beam is, so we can see it, rotate around, we can actually see it sort of generating like a cyclical motion of those. So if I stop this very rapidly, maybe. Okay, I'll change the direction. That works today. So you can see that the beads track it very fast. Let me do that again. So we're going one direction. Maybe I'm blocking with my hand. Now we're going to go the other direction. And now it's stopped. Okay, so if we wanted to scale this up to try and study how efficient this is, what would we have to do? Right, so this is like an experimental design question. So one, I'd have to fix the viscosity, right? Two, I'd have to make the swimmer neutrally buoyant so that I can drive it up and down correctly. So I'm gonna have to add some more weight. I'm gonna have to sit, and get it to tip. Three, we're gonna have to make sure that the motor is driving at an appropriately scaled rate. So we're gonna need to go to an expert in the motor and ask, like, what do we need to drive this at? And then four, we're gonna have to have some microscopy method that allows us to see this thing sort of turning in 3D and understand how fast are we both moving the fluid and how fast are we moving the swimmer, right? And then from all of that, we can start to build some picture of what this 2D plane looks like. But that's actually not really telling us the true flow because the true flow is in 3D, right? So if we want to do that, now what do we have to do? I have to automate my light sheet so it moves back and forth. I have to put the camera on a motor. I have to start trying to not shake this while I'm doing this because you'll notice if I shake it with a motor, it's going to move. And so all of a sudden, I'm starting to build what is a pretty interesting experiment. So when I showed Banu this, she immediately wanted to make a flexible propeller, right? And the reason is in nature, it's actually flexible. And so we have a lot of work to do on our side to try and make something like this work. But this is kind of what the classic fluid dynamics idea of this whole process is, right? Is to build this macroscopic model that we iteratively get closer and closer and closer to what we think the scaled up physics is. So we need to get the viscosity right, we need to get the swimmer right, we need to get all of these things correct. And so that's I would argue, a kind of non-trivial problem, right? This is tricky to take this scaled up version and get the thing that nature built, especially because this is a 3D printed swimmer. And so there's no flexibility in the tail, right? And so then we would have to figure out how to make a flexible polymer and do all these sort of things. So I hope this gives you a sense for, it's certainly possible to take these macroscopic experiments and try and learn something about what that biophysics that's underneath is, but it's a little bit tricky to know that you're actually measuring the real thing that you're interested in. So while I quickly wipe my hands with some of this water, are there any questions about this, the macroscopic model, or what I've said so far before I move on? Yeah. So this is a resin filament that is done in a DLP printer. Well, so the problem is you don't want it too flexible, right? So you need something that's close to the biology that has that polymer. So you need some blending, right, of how to do that. So I will say there's a group out of UCLA that's actually made a 2-filament sort of version of this where they can actually study the interactions of how the filaments couple with each other. And there they're using a version of a TPU resin that they homemade. And it was just straight trial and error. So, but yeah, so you could get there, right? All right, so now that my hands are not going to make everything incredibly sticky anymore. So I hope this at least motivates for you that while it's possible to study how this propulsion works on a macroscopic scaled up experiment, you know, what we really want to make sure that we can do is actually study the real thing. So what do we do to do that? So people have tried this macroscopic experiment a number of times. So actually Purcell proposed this in 1977 and proposed there was a way to drop a wire helix that was a scaled up version of the E. coli flagella, drop it in corn syrup like I've shown you here, and then measure how fast it's going down to rotate. And from that, there's a way to extract the hydrodynamic coefficients. I'm going to go into that more in a second. Rodenberg took the exact same idea I'm showing you here, but just used a flow cell and drove it to the right, and then tried to look at the resistance on the motor to understand how much was required there to balance things out. And then actually there's this beautiful experiment by Chattopadhyay where they optically trapped an E. coli and then asked how much force needs to happen for us to hold the trap there in place. So I'd argue here we're starting to get to that microscale, right? But we have to separate the effect of the head and the body here, which is something I didn't even talk about for the experiment that I've done. So here's what those look like. So if you spend a little bit more time with this model I just made and you make this, you can actually make the swimmer do this. So you can, and so the important thing I want you to notice here is actually the head is counter rotating to the tail. And so this is sort of satisfying these low Reynolds number fluid dynamics where we expect the whole system to be torque free. You know, you can drive this and they have this sort of flow cell inside of here and they're trying to look at it. This experiment is actually what inspired the one that we did with Evan this summer. I would say that this sounded a lot easier than it was at the beginning, but I'm extremely happy about how well it works. And so we have some pretty nice particle image velocimetry flow from this now that, you know, is actually telling us sort of how much are those beads inside there being displaced from this fish tank model that I'm showing you right here. So I just spent a little bit of time about hydrodynamics and propulsion. Let's come back to that whole beginning of the talk I gave you about fluctuation dissipation. So we talked about this idea that we can have a sphere. The sphere is going to have some velocity. We can think about there being a force on that sphere, and there's going to be, you know, some drag coefficient times the velocity. Also, there could be a torque, but we're never going to see that experimentally, right? Unless we manage to put a sticker on it and moved around, you know, and track that. Experimentally, that's a very hard thing to see on a sphere. There are other systems that rotate that one can think about breaking that degeneracy. And so that's actually what Purcell kind of hinted at here. Purcell was hinting at that because this is a, you know, a system that has this sort of omega that we should be able to measure, we should be able to actually do something very similar where we use what's called the resistance or the propulsion matrix to measure, let's say, if we have an applied force, which would be gravity in a solution, if we drop this, you know, vertically, to then figure out what these coefficients are. So there's a blueprint here that kind of teaches us about if we want to not have all these complications of the macroscopic experiment, we just want to isolate that propeller and look at it, we can do that. So what we worked on with Frankie and Peter in collaboration with Rizal and actually Bernard Yuki is we thought about how to do this straight from a fluctuation dissipation point of view. So we have some helix that we're going to cut off. We're going to cut the propeller off the E. coli. We're going to turn it into a colloidal particle. We're going to remove the actively driven biology part, and we're going to just let thermal energy do its thing. And so what do we want to do? Well, it turns out that there's a version of the Einstein relation, which Brenner derived, and I'm going to go through what he did in just a second, that tells us, okay, now we need to know the motion along every single one of these axes and their correlations to be able to figure this out. So we're going to get some tensor that tells us something about how this is, having its drag in the solution. And that's going to now be proportional to some matrix of diffusion coefficients that we're going to derive. So our job is to be able to measure this thing, basically do the experiment that Perrin did, but do it for a microscopic helix. Now, when I say microscopic here, there's only one dimension in this that's actually microscopic, and that's the length. The filament size is something in the order of, say, 20 nanometers. And then the pitch is, let's say, under a micron. I don't want to get too far into the dirty details about that. But the pitch, which is here, is about under a micron. The filament is 20 nanometers. And this is the ones that we cut off. We got something around 8 microns. That's, a piece of the flagella. So what we want to do is we want to do this, but we're going to need to change the way we do the microscopy a little bit. So again, our goal is to go from the sort of observations of Perrin and the theory of Einstein, take these things that Brenner very carefully derived about how to relate the resistance matrix, right, this tensor of sort of coefficients here, to the force and the torque, take all those ideas and turn this into, a fluctuation dissipation problem. We're going to sidestep that whole biology active-driven part of it. All right, so this sounds not so bad. I told you we make fancy microscopes, but there's a problem here. The problem is that using optical microscopy, we can only get down to a certain resolution. And so this week, I asked Peter to take one of the best microscopes we have in the lab and take an image of a 100 nanometer bead. And so under the microscope, due to the diffraction of light and just what's called the transfer function of the microscope, where we lose resolution as the features get finer, we get something that's about 240 nanometers in XY. That's the best we can do. And we get something about 650 nanometers in Z. And so what does that mean? That means that 20 nanometer filament that has a pitch that's about our Z resolution, we actually don't really have a chance of seeing it turn in solution if we use a standard approach to microscopy. And so just to be really concrete, this is actually data that Frankie took early on, where he put flagella. It actually kind of tethered itself to a cover slip. And I think Steve and I have had arguments about what you can statistically infer and not. I would argue it's quite hard to infer what pitch is what from this and which part of the helix is where. We could try hard. In fact, we didn't try that hard, right? So maybe we could have done better. But my argument is we should just take an alternative approach to the microscopy and do this in a way that it's going to work. So let's make a Z-stack, right? What do we do? If we have a microscope with a focal plane, let's bounce the focal plane up and down as fast as we can and make a 3D image. This sounds like a reasonable starting approach. I suggested it for the fluid. So you can buy really fancy piezo motors nowadays. They can move very, very fast. The problem is, if you want to just say cover 15 microns at the correct sampling to rebuild that helix, it's going to take at least 300 milliseconds, and that's assuming you're not noise limited in your camera. So actually, this is much too slow to image the rotation, the coupling of rotation and translation of these, even in a reasonably high viscosity solution. We need to do something on the order of 10 times per second, not sort of 3 times per second. The other thing is, if I take an actively driven object and couple it to a fluid cell and drive it very hard, I'm going to start doing what I did here when I banged on the glass. I'm going to start driving that fluid in addition to doing it. And I'm going to change the physics of the problem. So we need a low inertia way of imaging where we don't impart inertia, and we need a reasonable resolution. So this is a toy we have up in the lab. This toy is called the Mirascope. It was discovered at UCSB by a member of the facility staff when he was trying to clean 2 parabolic mirrors that were on top of each other. And the large parts of the mirrors were facing each other. He kept cleaning the dust, and it kept not coming off. So he went and he got Virgil Ellings, and they thought about the problem. And it turns out what they had discovered was that if you put 2 parabolic mirrors like this and you have an object here, you actually can recreate a perfect 3D copy of that object in air. So this actually satisfies the perfect imaging condition where we have a completely perfect copy. And let me play it again. We can look at this frog from any direction we want, and it's 100% the same as if we were looking at the actual object and turning it in our hands. This is really cool, right? So they patented this. This was patented in 1970. That actually was Elling's first patent. He went on to found Digital Instruments, which some of you know is like the scanning probe microscopy company, right? So what we're going to do is we're going to turn this idea of this toy into a microscope. So how do we do that? So the reason this toy works is the object's in air and the image is in air. We want to image in water. So it turns out that what we have to do is we have to build sort of a second microscope to undo the stretching that occurs from imaging in water that goes into air. And then we can image that again with a third microscope. So this is a process in physics called remote focus. And really one of the best first sort of demonstrations of this was done by Botcherby, Booth, and Wilson. It's actually relatively recent, right? We're sort of talking in the last 20 years. And so I just give you an example here of what they can do. Here is if you just took a normal microscope and you had a bead at the focus and you sort of tried to displace your camera or something away from your sample to limit inertia, you start to get these aberrations where your impulse, your point spread function of your instrument spreads out. This is the best the instrument can do. But if we use this remote refocus strategy, we can actually build perfect focusing over, say, 80 microns, right? And we're doing that by moving this imaging system over here, which means if our helix is over here, we never touch it. So we're actually able to move that. You can then further couple that with an idea known as light sheet microscopy. And so when I first came here, I invited Jan Huisken. He's one of the inventors of this. He came and gave us a seminar on the fundamentals of light sheet. I'm not going to go through them today. What I am going to say is the same idea I took here of shaping the laser beam into something that's tall and then focuses in one direction. But what we do is we actually do this with that remote focus idea so that we shine a tilted laser beam. And then actually we literally take a picture of this tilted plane through the optical physics of the device. And so this is a work that I did in collaboration with Andrew York, Al Militsiki, and Reto Fioca. And this is taken off now. So Leica's commercializing a version of this. Labs around the world are building this. This is sort of known as basically, it's probably going to replace the workhorse confocal microscope because it's much more photogenyl and it's much faster. For our purposes, the resolution's about as good as what I showed you before. And here we can make an image by moving this little mirror that has very low inertia and it's not physically coupled to where our sample is. So to be really clear about how the scanning works, what happens is we move this mirror and through sort of aligning everything correctly, and what you get is you get the ability to displace this tilted laser beam that's coming out, and you also displace the imaging plane, which is in black. And then the optics that are on the backside here undo that tilt so that you get a perfectly diffraction limited image coming out the other side. So this is called oblique plane microscopy, and it's made possible by this lens that we all nicknamed Snouty because we had to cut part of the glass off to do it. So this is a method that we've really worked a lot on in my lab, and this problem I'm telling you about today is sort of a perfect application of it. To build a whole lot of statistics of helices diffusing in fluid just like Perrin did. Except now we have both the just enough resolution that we can see that twist, right? And so now we're actually doing the same thing Perrin did. We're taking the same idea of using fluctuation dissipation to try and then learn something about this system, but we're going to try and go a step farther. We're going to try and learn something about the propulsion. So what do? We use a machine learning model to then segment where the bright pixels are against the background. And then what we do is we actually use a physical model of what the helix should look like to fit that to the shape of the helix. And from that, we can then stick a dot in the middle of it and very accurately track all of its diffusion in every direction. We can also get the correlations, and the important correlation that we can get out here is rotation and translation, right? Because if I have this helix, just like I showed you with our little demo here, if I want to move, I need to rotate so that I can have a translation. So if we know how efficient that coupling is, we know how good the propeller is. That's the long and short of this experiment. So after a fair amount of statistics and actually reworking some things which are good cautionary tales for students that I'm happy to talk about afterwards about measuring viscosity, what we found is that there's a negative correlation between rotation and the translation. And in fact, this is important because in nature, it's a left-handed helix. So we didn't pre-specify this at all. We just fit it to a helical model. And what came out at the end is that we actually got the right sign. And we actually see that the coupling sort of has the right scaling laws. We change the viscosity of the solution that it's in. And from all this, we can actually extract those A, B, and D coefficients that Brenner and Purcell taught us about, right, which are extensions of Stokes-Einstein. And you can get some of this big table. The point of this big table is that our work broadly lines up with what other people have done from these macroscopic experiments. But there are some small deviations. And in fact, this was a very interesting exchange with the reviewers during the process of like, where could these deviations come from? And one of them is probably this slight flexibility of the polymer that we're missing in our model. The other thing I want to point out is it's a bad propeller. So if we decouple the body and the propeller and we ask, okay, let's assume that the hydrodynamic quantities of that body stay the same, but let's scale that propeller using those to be the best they possibly could be and take a guess at how efficient this could be given the geometry. We get something on the order of like 1.7% efficient at turning that mechanical work on the motor into sort of forward, useful work. So this brings up a whole host of evolutionary questions, right? Of like, why would this occur? Like, why would nature select this process? I don't have an answer to that. I will say that Mike Lynch and Schaffmacher, who are colleagues here at ASU, they recently published this interesting paper where they surveyed the energy cost of building flagella across the tree of life. And one important thing I want to point out is building one of those fancier sort of ways of swimming like spermatozoa or protists use would cost nearly all of the ATP and even just all of the protein building blocks that are inside the bacteria to make one of those propellers. So this is maybe potentially some good enough situation, right? Don't actually need to swim and it doesn't actually cost that much metabolism. And so it's sort of good enough to get there, right? And so this is an open question. And so one of the logical next step, right, is to try and then go through and do this for lots of different bacteria and sort of start asking the questions about whether or not this is a universal observation that these propellers are bad. One can also try for the other types of propellers, right, and see if we can get the same results out of it. So, to me, this was a really just sort of beautiful application of sort of this biological question, right, coming together with this fundamental physics that met together in this realm of this statistical physics and fluctuation dissipation. It drove our interest in these swimming problems. And one of the things that, is not accounted for at all in these problems, and people tend to only have models of this, there's very little experimental evidence, is that when swimmers are near surfaces or when they're near each other, actually there are some hydrodynamic interactions between them, right? And one of the key ones of this is if a bacteria comes near a surface, to keep that torque-free condition, and remember I told you what that was, we have to have the body counter-rotating to the tail as it moves. When it's near the surface, it doesn't have free fluid underneath it anymore. It actually makes a loop to keep the torque-free condition. And this is solely governed, the thought is, by the hydrodynamics of the system, by the fluid dynamics. If you change the surface roughness, the direction of the loop changes. So actually, this is a beautiful example of boundary conditions, right? So if we change the boundary conditions of the problem, we get a different solution to sort of what's happening with Navier-Stokes there. So how do we test that? No one has visualized it. People have seen the bacteria swim. No one has actually gone in and watched particles move around these things while they're doing it. And the reason for that is the timescales in this problem and the resolutions required are quite high. And so this is new work that I'm going to show you now in the sort of last 5 minutes here, where we're really trying to take ideas from different parts of microscopy and particularly tomography, which I know a lot of people in this room deal with in a different context, and use those to try and image these systems fast enough that we can actually test that fluid dynamics prediction. Because again, there's a lot of modeling done, but there hasn't been an experimental validation. So how do we map the flow field? So in this demonstration that I had a second ago, you could see little bubbles and you could see the tracer particles in there. And as the tail moved, the bubbles moved around, right? So if we track those particles over time and we statistically build those up enough, we can actually get the fluid flow out. The thing about this system is it's pretty macroscopic. If we try to do this on the microscopic scale, actually the Brownian motion of those particles themselves is going to interfere with our ability to tell how much the fluid dealt with it. So what we need to be able to do is we need to be able to image really fast at reasonably high resolution. Not as high resolution as the flagella problem, but reasonably fast. The problem is there's no free lunch in linear optics. And what do I mean by that? I mean that if we want to go faster and we're using fluorescent labels, we need to turn the laser up. But if we have things that are living, it's very easy to make them not living anymore by turning the laser up. So there's this sort of triangle of frustration people talk about in my field, where sample health, right, has to deal with how fast we're going or the resolution we need, the signal noise. There's a photon budget issue. It's a photo damage and photon budget issue. And so what happens here? So let's take a couple microscopes, let's turn it up, and let's just watch how bright these cells are over 50 frames acquired at 10 milliseconds a frame. And hope everyone can appreciate the fluorescence is going away, right? And again, this is a byproduct of using fluorescence. And so for those of you who aren't familiar with it, there's a process by which the laser light's absorbed. You can then get a molecule that's in an excited state. When it goes back down to the ground state and emits a photon of a different color, that's what we detect. That's how we did the flagella project. And so you can actually quantify this and see that it's a pretty linear photo bleaching over time in this case. So one could run the laser lower and extract low signal to noise statistics, but then it's hard to run the detector fast, right? And what do I mean by fast here? I mean we need to be something on the order of like 600 to 1500 volumes per second, not planes, full 3D volumes to see these interactions, because we need to see the bacteria come near the surface, and we need to be imaging fast enough that we can tell what's Brownian motion versus the beads. So what we're going to do is we're going to try and get away from using fluorophores. So we're not going to do the thing that I did in this tank here. What we're going to do is we're going to try and exploit the fact that when light passes through a sample, it interacts with that sample. So some of you probably know this just from using your cameras to take pictures, right? Or if people have used film in here, you understand that interaction between film, right? You have some chemical reaction. So what happens? You have light that comes in, it hits your sample, light interacts with the sample, you have an imaging system, you then take a picture. So here you're just detecting the amplitude of the electric field at the end. You've lost any information about the phase. So one could build a hologram. This is another source of a large set of Nobel Prizes, right? So what do? We take one piece of the light, we shine it through the sample, we take a copy of that light, which maybe we make by using a beam splitter or a fiber splitter, and then we shine it in. We can do it on axis or off axis. There's lots of strategies here. Now we get something that's striped. Those stripes tell us something about the phase of the electric field, right? So now we've recovered information we didn't have before. But this is still a 2D picture. We can refocus it in 3D, but it's not inherently a 3D version of the object. So if we now start tilting the angle that the waves come in at, and then we do the same hologram, we get n holograms for n angles. We then have a mathematical problem, the inverse problem of, given a bunch of holograms that I observed, what is the refractive index of the sample, the density of the sample that I did? So this is known as diffraction tomography, and in this case, I'm describing optical diffraction tomography. This is not unique to visible light or IR light. In fact, Wolf has a very nice paper where he sort of fundamentally shows that any diffraction tomography follows the same rules. The actions you have to take to get there at the end are different. So, you know, for those of you that have thought about CT or X-ray computed tomography, you take an X-ray source, you spin it around the human body, and you get some X-rays. You then have to, put them back together, and you learn something about the absorption of the X-rays. The governing equation for all of this is the Helmholtz equation. For optical diffraction tomography, we change the angle that the laser beam is incident in, let's say, our biological sample. It doesn't have to be biological. It could be anything with a refractive index. We get out holograms. In the end, we're going to learn something about the refractive index. But again, the governing equation is the Helmholtz equation. Right? In both cases, the Helmholtz equation is governing how this works. So that says the propagation of light through the media is going to get us to the answer at the end. So what does this practically look like? It means we need to build a microscope that changes the angles, and we get holograms, and then we need to do some math to invert the problem. So there's lots of ways of going about this. We are learning a ton about them at the moment. I have a feeling that deep learning is changing the way that this is happening rapidly. But what I am going to say is you can think about doing this in the situation where you think you only have a single scattering event from the light, or you can try and sort of account for more complex samples that have multiple scattering events. For this bacteria swimming problem we're working on, we really don't need to worry about multiple scattering that much because we have tiny beads that are fluid tracers, and then we have pretty small bacteria that are swimming. So in fact, we can just reconstruct this in the normal way. And then what we're going to do is we're going to use these devices called digital micromirror devices that we spent a lot of time in my lab thinking about how to use in these situations that have some unique diffraction physics on their own. So with those, we can actually change the pattern that we put into that sample at 10 kilohertz. So we can shoot light and constantly be changing the angle actually a little bit faster than 10 kilohertz. The thing is, you have to buy a very high-speed camera. So we now own one of these like sports slow-mo cameras, where you see people like shoot bullets into wood or slap each other in the face and see how your skin changes. So we've integrated one of those into our system, and just some early proof of principle experiments, we've tracked beads sort of going slow, 6 volumes a second. We've looked at denser samples, so we've ramped up the density. More importantly, we've done experiments sort of at 650 volumes a second, where actually you can see the Brownian motion is frozen. So what I'm showing you here is the XY, XZ, and YZ projections of the data. And you can see in all 3 versions of the 3D data, we're taking 650 of these snapshots a second, and we're going faster than Brownian motion is because these are the same beads that are over here. So the wiggling and jiggling of those beads in water, and again, this is in water now, no longer a high viscosity solution, we can freeze that. So we can go fast enough that we can tell whether or not it's Brownian motion or whether or not it's fluid dynamics. So an interesting experiment you can do with those beads is that you can ask, as we approach a surface, what happens to the diffusion, that we see? And one actually sees that as you approach the surface, there's some interaction with the surface where you no longer have the freedom to diffuse the way you want to, and you actually get a drop in the step size that the beads take at any time point based on how close they are to the surface. And you can fit this exactly to an analytical model that's an expansion of what you would expect from the hydrodynamics. So we've really built this tool up, which again doesn't have the resolution of the fluorescence experiment I showed you in the beginning. Our resolutions aren't quite as good, but our timing resolution is outrageously higher compared to that flagella work that I showed you. We're talking two orders of magnitude. So the early data we have, and this will be the last piece of data that I show, is that we've done the early parts of the tracking experiment. So the darker red lines are actually E. coli that are swimming around from Navish's lab. The brighter little dots are the tracer particles. And what we're showing here is that we're actually tracing these things in 3D as they're swimming around. And what we're working on now, sort of how do you quantify this and put this into a statistical framework so you can actually generate the average fluid flow fields around these bacteria as they're swimming near surfaces, right? So there's a huge amount of sort of modeling and image processing that have to go into this. But this is where, this project is pretty much working now. We only had to go at 150 volumes per second here. We have some interesting ideas about how to go faster. And those involve multiplexing beams. And I think some people in the room who work on, the X-ray tomography or sort of E-beam ideas have thought about this too. I'd be super interested in talking more about this because this is a world we're learning a lot about. And so what we've done is we're actually shooting multiple patterns in at once. And then we're showing that if we just go one pattern at a time, we get this pretty nice data, but we can go all the way to 19 patterns at a time and still back out something that looks reasonable. So we can get another 19 or 20 speed up from how fast we are already going by using these multiplex patterns. All right, so in summary, fluctuations and sort of thinking about these things as inverse problems, right? I measured something, how do I understand the physics that caused it? In my opinion, this can work on complex biology, which I didn't show you any sort of work on, but I will say we spent a lot of time on that and it works extremely well. And sort of molecular biophysics, right? Things that are working there. Both molecular motion and microscopes, they're all governed by physics. And so in my group, we really think about both the physics of light, right? How do we make better instruments, but then also what happens on the outside. And we're really excited about this tomographic phase imaging because it's breaking what's called this space-time bandwidth that fluorescence microscope has. That means how many things can we image per second at what resolution, right? This is really getting us higher. All right, so there's a huge amount of people that have done this work. So I'd like to first off thank Peter Brown, who's done an incredible amount of the work that I've shown you here today, along with Frankie and Rizal, as you mentioned. And so they were integral to doing this flagella project. We worked on it together. I would say there's a huge part of my lab that wasn't represented here at the moment. And so the people in that are working on these various parts of these other biophysics problems. I actually had a number of good PhD students and postdocs at Anschutz that did all this early work on light sheet microscopy and molecular labeling. In particular, Megan Ahern, who's now actually down at University of Arizona. And then, we have a number of collaborators both here and elsewhere. And this work has been, funded on and off by NIH. But I would say recently, Chan Zuckerberg Initiative has really stepped in and fund people who are interested in fundamental imaging science. So if it is an area that you're interested in deep tissue imaging, or, other applications that are going on there, Chan Zuckerberg is really funding these with the sort of caveat that you make your work open. So you have to think about what that means. And I will say Knowledge Enterprise actually is not very happy about the terms of the grant sometimes, but they are supporting fundamental imaging science, which is lacking at NIH and NSF at the moment. It is very hard to get work on new optical microscopy done, right? This is a tricky thing to get funded. All right, so I think that's all I have, a couple backup slides, but I'll stop there. Thank you.